Hi, welcome to the Bioinformatics Chat. Today I'm speaking with Trang Lê. Trang is a postdoctoral researcher at the University of Pennsylvania, and she recently published a preprint titled Statistical Inference Relief Feature Selection. Trang, welcome to the podcast.
Hi, thanks for having me.
Trang, let's start with your scientific journey.
So back in 2010, I came to the University of Tulsa, Oklahoma, to study geology and math. And as an undergrad, I took a networks theory course and then got sucked into all these neat linear algebra tricks of eigenvalues and eigenvectors, to compute the different network characteristics. And the professor of this course, Brett McKinney, is who's actually a theoretical physicist by training, but he's done a great deal of research in bioinformatics. And so after several small bioinformatics projects with Brett, it was a natural transition for me to move into this field. Brett later became my PhD advisor and mentor and also running partner. And I should mention that Brett's the PI of this STIR study as well. And so after, I guess, about 6 years, I obtained my PhD in mathematics. And I had just moved to the University of Pennsylvania this year to begin my postdoc study.
Cool. And what did you do in your PhD, and what are your plans for the postdoc?
Right. So during my PhD years, I studied the overlapping between differential privacy and generalization in machine learning, actually. So with what people have done with differential privacy is to use it as a definition to sort of protect individuals' privacy in these large data sets. But what I do is I take that concept into machine learning and trying to protect the privacy of each individual single data point to prevent overfitting of machine learning methods. So that's what my PhD thesis was on.
Interesting. So that's not, that doesn't sound like anything sort of bioinformatics related. So I'm curious how you got to write about, differential gene expression, which I think only superficially relates to differential privacy.
Right. Yeah. So differential privacy was a big part of it, but I wanted to apply it to machine learning. And so during my time, doing all this research in machine learning, I stumbled upon a lot of machine learning algorithms like relief and, of course, random forest, gradient boosting, all of that. And actually, in my thesis, I compared, the performance of different feature selection and classification methods incorporated into differential privacy. And so, the application that I used was on neuroimaging data and also gene expression data. And that's how they all kind of tied together.
And for your postdoc, will you continue just working in general machine learning, or are you moving to biology?
I'm going to stay primarily in machine learning, but obviously my focus is on biological application. And right now I'm moving a little bit toward automated machine learning. Particularly TPOT. It's a tree-based pipeline optimization algorithm that helps scientists to not having to go through all these tests of which classifiers, which feature selectors, which transformers I have to use on these data. So it helps automate that process and eventually give you a pipeline of okay, you're going to use these features, you're going to use these transformers, and then you're going to use this, say random forest classifier, and that would give you the best cross-validated accuracy on your training set, for example. And so I'm trying to help with that automation process.
Very cool. Our today's discussion will be focused on your paper about something called STIR. That's your statistical inference relief algorithm.
Yes.
And the context is feature selection, which could be applied to genome-wide association studies, could be applied to differential gene expression studies. And the need for it, I guess, is driven by something called epistasis in the context of GWAS. So maybe let's introduce this. What is epistasis?
Epistasis is essentially an interaction structure of genes that affect a phenotype or a biological outcome. And that's to my understanding. And the idea of epistasis has been used most often in the context of GWAS, the genome-wide association studies, to find polymorphisms, the SNPs, who are interacting with each other and also affecting a biological outcome. But in this particular paper, and what I've been using, epistasis is applying it to gene expression data.
Right. So maybe one way to explain epistasis is that when we look at the same locus on two different chromosomes, then we have potentially two different alleles. And the way these alleles combine, it could be additive. So their effects of each allele, they're just added. Or there could be non-additive interaction, which is, for example, dominance, when one allele suppresses the other one, right? So the way I understand epistasis is the same, but with multiple loci. So when one locus can act as sort of dominant or recessive in relation to another locus.
Yes, that's exactly the traditional way to think about it. In the context of gene expression, it's and I'm going to do this very roughly because I'm not a biologist, but essentially, if one gene is downregulating another gene, and somehow this downregulation affect the outcome, as in, whether you're sick or not sick with a particular disease, then we want to be able to find that downregulation in the final pipeline.
Yeah, and the models that are usually used for GWAS or for differential gene expression, right? They typically don't do anything to account or correct for these interactions. So if we're talking about GWAS, then we could use, for instance, linear models to model the effects of different loci, and we had a podcast episode about that. And if you wanted to account for epistasis, what would you do? You would want presumably to include your interaction effects in your linear model. So you would have main effects for each variable locus, and you might also include the effect of two loci interacting. But the problem with that is that leads to an explosion in the number of tests that you will be performing.
That's exactly correct. Yeah.
And so what is your solution to that problem?
Right. That's exactly our motivation, not having to explicitly write down, those interaction terms, because in the case of, say, in gene expression, you have thousands of genes, then you would have, a combination of how many times you can choose two of those genes out of these thousands to write down all those interaction terms. And then, like you said, it would become an explosion of tests. And so this is where relief comes in.
In general, right, we need some way to select our feature. So I suppose that's where the feature selection algorithms come in. And so relief is one of those algorithms, right?
Right. Yeah. So in like you said, linear model is one of, among others, feature selection methods that assume this independence among the variables. Other methods include information gain or the gene index. But to be able to find these interactions without explicitly writing out these terms, we want to find a feature selection algorithm that considers the relationships between the variables, and also we need to do so in a computationally efficient manner. So, and relief is one of those. Yeah.
Right. So the idea is that let's say we have 1,000 covariates. So like in a gene in a differential gene expression study, let's say we have 1,000 genes or more like 20,000 genes, but let's say it's just 1,000 genes, and that will be 1,000 main effects, right? And then if we wanted to include all the interaction terms, there will be like 500,000 more terms,
Correct.
which is just too many. So the idea is that we use this feature selection algorithm to rank all the interaction effects and say, okay, out of this 500,000, we'll select maybe another 1,000 interaction terms and include them in the model.
Correct.
Here's what I'm interested in. Like then, typically, once you include these interaction effects into your model, you'd usually compute some p-values, right? You'd apply like multiple testing correction. But if your p-values come from a features, if your features come from a feature selection algorithms, wouldn't that confound the multiple testing correction, because you're sort of pre-selecting them, right? So your p-values would be artificially low just because you selected the low p-values in the first place, or selected these very salient interaction effects, but they would be no longer sort of random and independent.
That's exactly right. And that's sort of like the double dipping issue with our data. If we pre-select these features and then we want to say apply it or find the p-value again on the same set of data, then we would have a very optimistic view of our p-value. Right now it's very low because we have pre-selected them. And so and I think it's very interesting to think about statistical inference and then machine learning, because each field, I guess, almost have their own objective toward, do I look at the p-value, or do I want to create a model that give me a very high accuracy at the end? But to be safe and to make reviewers happy, I would say, if you really need to come up with some sort of model to provide the accuracy, then choose a different data set, an independent data set to build your model. But I would like to also add that the 5 or 500,000 interaction terms that you were talking about, that only consider two-way interaction.
Yeah.
Now, if we go to higher order interaction, it's going to become even more complicated. Yeah.
So is that how it's called, double dipping? This is the first time I hear about it, but
really?
I like this name.
I think that's a casual term to call it. People have called it p-hacking. Yeah, so I try to avoid using my label twice. Although some people have different idea and they say, well, I already have this significantly associated with phenotype features, then why can't I use them to build my model? I just need a model out of this. I think if you just need a model, it's fine. But if you claim that your model has a particularly high accuracy in classifying individuals, then I think you need to definitely test your model on a different data set.
Right. Or if you claim some causal interactions, right? Not just predictive efficiency.
Exactly.
And of course, your work is cool in that it allows to compute p-values inside the same process, right, as feature selection. So those are not two different processes. That's just a single algorithm, and so that should avoid that issue, perhaps. But we'll get to that. But I think it's a good time to introduce Relief in general. Like, what is it? What problem does it solve? Oh, I guess we already know what problem it solves, right? But how does it solve that problem?
Sure. So Relief is a family of machine learning algorithms that select features, or I think in the original algorithm they called it these attributes, that associated with the your outcome of interest. And by using nearest neighbors, it's able to find the association that may be due to not only individual effects, but also epistasis or statistical interactions with other features. So, it takes into account the relationship between these attributes.
Yeah, maybe we should also explain, like, if people are not familiar with this term feature, but that basically means, yeah, like one of the attributes or covariates. So for GWAS, that would be an allele in a certain locus, and for differential gene expression, that would be
The
expression
expression
level of a
level,
certain gene. Yeah.
That's exactly right.
And so we want to select those features, so either genes or loci that are responsible for the certain phenotype that we're observing.
That's correct. And by nearest neighbors, I mean this is among the sample space. And so if your gene expression level is very similar to, say, your parent, one of your parents, then that person would be your nearest neighbor. In a way, in finding the neighborhood of a particular sample, it takes into account all the attributes, all the features, and not just one, even though in the algorithm itself, the feature score updates is only one at a time, because you're comparing your feature with your neighbor. It does this in a very high dimensional way, not just one at a time, like most people think.
So maybe we should just explain how the algorithm works, because I don't think it's that complicated as it may sound.
Yeah, sorry if I made it sound complicated, but yeah, you're right. It's not complicated at all. So basically, like I said before, you're going to find neighbors of each sample or observation, or in the original algorithm, they call it instance. But let's just call them sample for now.
Let's concentrate on one. Do you want to pick GWAS or differential gene expression, which is closer to you?
Let's do gene expression, if that's okay.
Yeah, let's do gene expression. So we have a certain number of samples, right? And
right. So we have a certain number of samples, and let's just consider 2 genes for now. And so if you imagine you have 2 genes, one in the x-axis and one in the y-axis, and these samples are, in the 2-dimensional space, then pick a particular sample, unify its neighbors, and then a hit is one that has the same label as your sample, and a miss is a neighbor who has a different label than your sample.
Yeah, and the label is just the phenotype. So if you consider like maybe healthy and sick patients,
that's correct.
Right? For instance, so you would label each point in this 2-dimensional plane as healthy or sick.
And so if you look across the if you just compute the x-axis difference between your sample and a miss, and a sample and the hit, then the algorithm updates the score of that gene X by adding the difference in that gene X attribute between your sample and a miss, and then subtracting that difference between your sample and the hit. And the reason for that is because you want to add the difference between your sample and the miss, because that gene X is meaningful in that sense. It's meaningful in differentiating the label of your sample compared to a miss. However, if there's a lot of difference between that gene X expression between the sample and a hit, then it's not that meaningful. So you want to subtract that.
Right. So the idea is that we want, so the good attributes, so for example, the good genes would be very close. So the difference would be small for samples in the same category. So let's say for healthy samples, it should be very close among all healthy samples, and it should be very close among all sick samples. But when you compare healthy samples with sick samples, the difference should ideally be large, and that would mean that this gene is meaningful in this study.
That's a perfect description of relief. Yes.
Yeah, and then you basically go through all the genes, you go through all the samples, and for each sample you consider either the closest neighbor or several closest neighbors, right? And at every point you update the weight of the gene using this procedure.
That's correct. Yes. And the number of neighbors is also one active field in relief. People are trying to find what is the best number of neighbors to be able to to detect these interactions. And so simulation studies have been done to find this. Originally, it has the default is K equals 10. So you use 10 nearest neighbors, no matter what your sample size is, which is very arbitrary. But recent papers have shown that, perhaps you can find an adaptive radius for each of your samples so that the score updating is more meaningful.
Yeah, and if I'm not mistaken, in the very original relief, it was even one, wasn't it?
That's correct, yeah.
Yeah, so they considered just one nearest neighbor, and then people thought, well, why just one? Why not take multiple? And that would sort of perhaps lead to more robust estimates when you average over several ones.
Yes, that's exactly right. And that the interesting part is that if you increase the neighbor radius to be infinity, for example, which means you're including all the samples, then the result is very similar to a t-test. So you, in a way, you're increasing the ability of relief to detect the main effect when you increase the radius. So that's that trade-off that we have to think about.
Right.
But
yeah. And on the other hand, if you consider just a few nearest neighbors, then you boost the effect of these interactions that we're interested in.
Right. Yes.
So relief has been around for a while, right? It's a rather old algorithm. And you don't hear about it all the time. Like all those people competing on Kaggle, they don't use relief that much. So do you think it didn't catch on?
Yeah, and I'm not sure why particularly. I'm hoping with more automation, by which I mean, these adaptive radius that people are trying to come up with, that you don't have to do a lot more testing around, see how many neighbors you have to do, you have to use for this algorithm to work. And yeah, it's interesting because in a way, and I think in the biological field especially, because we have so much noise, that I think algorithms like relief is really helpful. But perhaps those data on Kaggle, it's not as perhaps computationally expensive. Or my understanding is that, with GWAS and gene expression studies, that there's just so much noise out there, and there's only a very few signals, if you may, then relief becomes very effective. And there have been studies on, comparing relief with other algorithms. But yeah, I'm not exactly sure why. I'm hoping that with, these, and I'm gonna throw it out there, is the name is multi-surf is one way that you use to find the number of nearest neighbors. And so I think I'm hoping that with multi-surf coming out and more automation and stir, people will be able to use it more, and hopefully that would catch up at some point.
Yeah. Sometimes it's just fashion, right? It's just not fashionable. Like deep learning is fashionable, obviously, or this random boosted trees are fashionable. And if someone just comes into the field and sees that, this relief has been around for a while and no one uses it, then maybe it's just not that good. But I think it's a very nice algorithm. It's very easy to understand. It's almost obvious in retrospect, although, there are many of these things that once you learn about it, is this all? Is this it? Right? This is very trivial, but
right.
It's effective at what it does and very easy to understand, which is, I think, very cool.
Thank you. Oh, and another thing that I'd like to add is that it's still a feature selection method, so it doesn't have a model. It doesn't give you an accuracy in a way, and so oftentimes you have to incorporate it with something like you said, gradient boosting, to give it a score. How good did the algorithm perform? And so it's hard to judge whether the feature selection methods work if you don't have, an underlying structure of your data known, by which I mean with simulation study. And so these data that people use on Kaggle and stuff, it's more, perhaps some team tried to use some feature selection method. And I think most people do now, and transform the data set. They don't really show that at the beginning, but then at the end, this is, we eventually use XGBoost to get this accuracy. So
yeah, this is a very good point. I think that in machine learning, as we discussed earlier, you care about just predictive accuracy. So
right.
if your algorithm can cope with that, you'll just throw all the features into it, and then if the algorithm is good, then extra features, even if they are mostly noise, they won't hurt, and you just apply some regularization or cross-validation to avoid overfitting. But like with a good algorithm, extra features don't hurt. On the other hand, in statistics, again, as we discussed, you have this double dip problem. I like it so much, where it's hard to apply feature selection and then a model, because then the p-values will be confounded. So it looks like, even though the algorithm itself is cool, it's like both of these areas like almost don't want it for these two different reasons.
That's exactly correct. And I think that's a very interesting nuance between machine learning and statistics itself, even though they really coming together nowadays.
So one thing we could compare relief is, for example, random forests. Although random forest is not just feature selection, it can be actually used to perform regression or classification, but it could also be used to select the features. Right? So maybe if you could briefly introduce a random forest and then compare them with relief.
Sure. Yeah, so I do know that random forest is one of the most popular methods for classification, like as you mentioned, and also feature importance ranking. And they use, I think the original algorithm uses the Gini index to rank the features. Nowadays, people also use permutation, and I love random forest. It's a very nice algorithm, and I love the out of bag sort of along the way computed accuracy. But what we have found is that when we have only a few features that are interacting with each other, and there are so many background variables or noise, if you may, then the random forest has a harder time to find these interactions. And it once again depends on the number of features that I'm trying to think. In R, it's the m-try parameter. The number of features that you allow random forest to choose as a node at each time in its decision tree. So we have found that it has limited power to detect gene-gene interaction in high-dimensional data, especially when there are not that many signals.
But do you have a sense why?
So as I mentioned, it depends on the number of variables that it's selected for each node. And so I think the default is square root of n, or square root of number of features in total. So if you have 100 features in your data set, then random forest at each time in deciding which node it would select 10, it would try to select 10 random features among 100 of these, and then it tries to find the best one out of those 10, which is the core of random forest. That's where the randomness comes from, and it really helps with reducing the variance among all the trees. But at the same time, because these 10 features are randomly selected from the original 100, among the 90, maybe if your signal is among the leftover 90, it's hard to get these features out, and maybe only in one of these 100 trees that random forest is building that the signal is there in the node. And of course, you can increase the number of this square root of n to say n over 2, or even all of the features, but then that would decrease the ability of random forest to detect the ones. So it basically, what a random forest is doing is averaging all these decision trees that it's making. But if you increase this number to all the features to n, then all the trees are going to look very similar to each other, and it sort of lose that ability of averaging, of decreasing the variance amongst these models. So,
That's interesting. And I have a somewhat related observation. So it seems to me that for relief, it's actually not that good that, in order to locate the nearest neighbors, you're using all of the features. I think it might benefit from like randomly selecting subsets of features. And here's why. So in a in a highly dimensional space, we know that the distances, right, they mostly become the same. Like it's very hard to distinguish close neighbors from far neighbors in a many dimensional space, because you sort of average over or you sum over many coordinates, and everything sort of blends together. And especially in differential gene expression, where the number of samples is typically very small, like you could have 10 samples, let's say, in each condition, and compare them to 20,000 genes that you're considering. So would it be better if, instead of considering the full 20,000 dimensional space, you would maybe add like a third loop? So the relief algorithm already has two nested loops, one over samples, another over attributes. Then what if you added a third loop, which, when you figure out which are the nearest neighbors, you look at them not over all the dimensions, but maybe some subsets of dimensions to cure that curse of dimensionality?
That's exactly, and I just learned about this recently also. There is actually one group who's doing this. I don't remember exactly quite where. I want to say Princeton, but they actually building algorithms that finding neighbors based on a randomly selected subset of all the features. And I think people are still experimenting with this. I think it's a great idea, and especially in, when you have a lot of features and you want to reduce your computing time, then this would help a whole lot. And in a way, like you said, it would, you wouldn't have to consider all 20,000 features, but only a subset of that. And perhaps that would be more robust, especially with the small sample size that we often observe in bioinformatics in general. So no, that's a really great, valid idea. And some groups are experimenting with it.
Cool, because I saw a lot of variations because the core algorithm is so simple, right? There are so many different variations. Let's vary this parameter. Let's compute this adaptive number of neighbors. There are all sorts of generalization. But I was surprised when I never saw this obvious problem that in a highly dimensional space, nearest neighbors maybe don't work as well. Okay, are there any other generalizations or variations besides your own that we should talk about?
Ooh, I feel like there are, like you said, there are many, people have tried this relief. Of course, there's all the versions from relief A to relief F, and relief F is what people use most often, just because I think at the time it's the most generalized. It was applied to missing data. I'm not sure about missing data, but it was applied for a different type of endpoint, different kind of variables or features. And then there's relief seek. It's also adaptively choose the number of neighbors. So yeah, surf, turf, most of it is actually from, and then of course there's multi-serve, which is the primary algorithm that we apply in STIR, is to select the just different ways to select the number of features. The number of nearest neighbors for each sample, and there's a version of iterative relief as well. In which people, find the best features, and then they cut off the last 10 or so, and then they do it again with only those features, and then they iteratively do that until they get to a number of features that they like to keep in their final algorithm.
Okay, should we talk about multi-serve for a little bit? What's the core idea there?
Sure. So the idea here is actually also very simple. You compute the difference between a particular sample, the distance between a particular samples to all its neighbors, and actually all the other data points, all the other samples, and then you average these distances and take that average and then you subtract. Okay, let's stick to the simple for now. So you take the average of the distances from one particular sample to all the other data points, and then that would be the threshold at which every other samples that are less than that distance away is considered a neighbor. And so that's the idea of multi-serve. Now it has an additional parameter in there that the threshold is actually not just the average, but the average subtracted by the standard deviation of all the distances. So a slight different than just the average, but that's the core idea. So each sample will have a unique radius and so K is not just 10, but it varies.
Right. So instead of fixing the number of neighbors, you're fixing the radius and consider everyone within that radius as your neighbor.
Right. And what people have found with their simulation studies is that multi-serve was able to identify it's probably the most flexible method among all of these, surf and turf and even surf star and this multi-surf star as well, in the sense that it's able to detect main effect along with two-way interaction effects, through interaction effects, and even higher order. Now, of course, each algorithm has its own advantages. So, for example, multi-surf star can detect two-way interaction really well, but it just lost in the main effect. And so, I think multi-serve is a good intermediate, algorithm that we're able to detect most of the things that we've seen in simulated data.
Yeah, and then how do you actually detect interactions using relief? Do you have to explicitly consider all the pairs and all the triples, consider them as attributes, or how do you do that?
So it's sort of automatic. So when we compute the neighbors, when we compute the distance matrix, that's when the interaction is taken into account. So we don't really specify that, okay, if I'm a two-way interaction or if I'm a three-way interaction, we don't know that. We built that in our simulated data, but we don't have to tell Relief to do that. It's sort of automatically doing that by computing the distance matrix.
That makes sense. I see how that would, these interactions would affect the score of a feature, like of a gene, right? So the genes that interact with other genes might get a maybe higher score. And let's say you use these scores to select your features, you select top 100 genes, but that doesn't really tell you then which interactions to include in your downstream model.
You're exactly right. And that is one limitation of this algorithm. It just tells you that, these genes somehow are interacting, but we don't really know exactly, which one is interacting with which and how many ways are they interacting with each other. And that would have to be done in a separate step when you perhaps use these features to build your gene co-expression network later on. But yeah, Relief doesn't tell you that, and that is definitely a drawback.
Okay, so now we're coming to your paper, your preprint, and what was the problem that you tried to tackle? Because there are so many variants, they must cover everything, right? Everything you would want. There must be a variant of Relief out there that covers that. But what was your specific problem that you tried to solve using STIR?
So with STIR, I wanted to find a not so arbitrary cutoff for the number of features that I want to keep in my model, in my subsequent models. And what people have done so far is when they have a ranking of all these features, they choose an arbitrary cutoff. So they say, okay, I'm going to just focus on the top 100 features, and I'm going to take these, I'm going to put it in my model later, and I'm going to, perhaps build a network or use it in a classification model later on. But there's not really a way to find this cutoff, so it's arbitrary. So what I was trying to do here is to impose a distribution, if you may, a pseudo p-value to these rankings, so that I can say, okay, if you, and I'm not a big fan of p-values, but it is helpful. So if someone comes in and say, okay, I want to find the features that are associated with these, with my particular outcome, and I want my p-value to be 0.05, then, with STIR, you can compute a p-value for these features, and then you can say, okay, all these features that have p-values less than 0.05, those are the ones that you want to include in your model later. And so, in a way, I'm trying to bridge the machine learning side of it, which is Relief, with the statistical side of, other algorithms such as ANOVA, or like you said, just t-test together, so that I can have a p-value from a pseudo null distribution.
Right, that makes sense. But so you're adding pseudo like in many places, so you're hedging against something. What exactly is the issue there?
The issue here is that, so if you don't mind, I can back up a little bit. And what I've done in this particular STIR study is that I rewrote Relief F in a way so that it's more mathematically approachable, if you know what I mean. So instead of very computer sciencey doing the for loops, I'm rewriting it so that now the Relief score is just a subtraction of two vectors, and then I sum all of them up. But instead of using the original Relief by just subtracting, now I'm taking that difference and I'm dividing it by the pool standard deviation, and I claim that to be my t statistic. However, we have to note that because these scores, these differences that I'm subtracting, they are differences among the samples, and because they're neighbors, there are correlations between my observations in this pseudo t distribution, and that's where the pseudo comes from. I'm not claiming that all these samples are independent of each other, because the difference are calculated between the neighbors, and so it would for a distribution to be truly a distribution, then your sample has to be independent for you to apply statistics on them.
Right. So you're applying a t-test, which is based on a t-distribution, although your test statistics is not like, it probably doesn't follow the t-distribution exactly, but it's like a good enough approximation. And then you just compare these p-values with p-values from a permutation test, and you show that, like, in practice, they're quite similar, so the deviation probably doesn't matter too much.
Exactly. It is a complicated problem. And we decided to go with, and this is, I think most who have done, is just trying to test it out and, compare it with something that we know is correct. In this case, the permutation. And, permutation is very computationally expensive. And so we prefer not to do that. But for the purpose of comparison, we wanted to see how well it performs with permutation, and turns out that it performs really well. Now, if you're worried about this independence among the samples, then there are methods to tackle it. We thought about subsampling. We thought about, perhaps, I'm trying to remember what we did. We could do some variance regulation to adjust for this independence. But what we've seen is that just using STIR multi-surf, it performs on par with permutation. And so we stick with that. But yes, future works could be done figure out what's the optimal way to compute this p-value.
Yeah, and I like this a lot. I was very impressed because this is one of those things, yet another one of those things that seem obvious in retrospect. Like once you reformulate this problem, as you said, once you go from the for loops into like this is a difference of these two values, right? It's almost obvious to maybe apply a t-test because we're comparing two samples.
The mean, exactly.
Yeah, but I'm sure it wasn't nearly as obvious before you're shown this rearrangement of the terms.
Right. And I was pretty happy with that. And yeah, so I would say the reformulation really helps, me see that, this could be done in a different way. But also, we're sort of suggesting a different way to calculate the relief F-score, even though if you have dig into it, the supplementary materials, it's what we have computed is the score itself, or what we call this t-statistic. It's very similar to the original relief, even though we have divided this number by a pool standard deviation. It's not just the p-value, but we're also proposing a different way to calculate the score by dividing the difference.
Yeah, that's another interesting aspect of this work is that you divide by the standard deviation. So just to visualize this as far as it's possible in a podcast, so you calculate all this intra-class differences and inter-class differences, so this miss differences and hit differences, and then you compute the variances of the distances between samples in different classes and between samples in the same classes. Now, would it also make sense to compute different variances for each class, maybe? So, like, you could compute just one variance, just consider all the differences and compute a single variance. Now, you propose to compute two different variances, one intra-class and one interclass. But then you could go further and say, well, we have two different classes, we have two different conditions. There's no reason to assume that those variances are the same. Would it make sense to compute like maybe four variances?
That's very interesting. I haven't thought that far. Yeah, I would think about that.
Okay.
I wonder how different it would, what the difference it would make to compute all those variances. But for when I first thought about this, like I needed to write this up right away because it's really cool. But yeah, I definitely think about that. Thanks.
Yeah, and then there is the question of like generalizing to multiple classes. So you tackle the problem for just two classes. But of course, if we have more than two classes, then maybe, like still a question of like how many variances you compute and you would need to perform the sort of pairwise t-test or maybe something similar to ANOVA then. I think you mentioned something like that, right?
Yeah. So this could be extended. That's, there's a lot of works in the future will be done. It could be extended to multi-class, like you mentioned, or even continuous endpoint type of thing when you do regression. And so right now STIR is applied for continuous variables. So in this case, gene expression level and binary outcome. But to apply this for GWAS data, we would need to do a slight modification on the diff function to incorporate in categorical variables in case of GWAS. Yeah, so there's work to be done, but I'm excited about the result, and it should not be, we just have to do a little bit more testing and spend some more time in making sure that, the algorithm doesn't break if I have a missing data point or something. So, but it should be rather straightforward. And even to incorporate this new way of calculation into existing packages. For example, my one of my co-authors in here, Ryan, he has on GitHub a package called Rebate and Rebate Scikit for the Python version of it that also calculates the relief F score with different variations, as I mentioned, multi-surf. So he's going to modify his calculation in Rebate to calculate stir as well. Yeah, so it could be, this basically could be adapted, I guess is the word, to many of the existing packages that's out there right now.
Right. So you mentioned a lot of future plans. Is this something you plan to continue doing yourself, or are you leaving this up for grabs?
I don't know how much time I have to do this with many other things that I'm having to work out right now. But it's definitely up for grabs if anyone's really interested. And I'm trying to make my code and everything as transparent and as available as possible. So Stir is actually on GitHub right now as an R package. And anyone could go in and dig into the algorithm. And I welcome any contribution and also criticism. I'm a mathematician, so my code is probably not as most elegant as most people would consider. But so it's on there.
Okay, and so what would the workflow look like step by step, if you are trying to analyze, let's say, a differential gene expression experiment? So let's say you have two conditions, some number of samples in each condition, and you measure the gene expression for each gene, for each condition. So you have this huge matrix. How do you then apply stir and maybe some downstream analysis? What would this look like?
That's a really good question. I think it's going to be practical. So if you have, let's assume you have a gene expression matrix with M number of samples and P number of genes, and you have two conditions, which means you have labels for each samples. Then really simple in R, you can load in your data or your data frames in this case, and you run and you just do stir. And then you feed into the matrix and then you tell them what the labels are. Then what Stir would do is, in the case in a real data set of 5,000 genes, approximately 5,000 genes and 200 samples, it takes about 20 seconds to run. And it would give you the genes. Well, it would give you the p-values of all the genes, but I think the output that you would see on your screen is the genes that have p-value less than 0.05 that are associated with your label. And so from here, you can test these genes on an independent data set that you have, build a model from it, and then see, once again, if you're interested in accuracy, you want to see how predictive these genes are in classifying these labels. Hopefully it's good. Or you can also build a gene co-expression networks out of this, perhaps using the interaction to the score that you have seen already, or perhaps even existing literature. So you can put this in, say, IMP. If you have heard of it, it's a software that Princeton builds for analyzing genes that you can put in a set of genes and then it'll give you how these genes interact from existing literature.
So these p-values that Stir outputs, are they already corrected?
Yes, these are FDR corrected p-values, yes. But it also, I think, gives you the raw p-values as well if you need them.
Right. So you could use these selected genes for a downstream model, so maybe to build a network or do some other analysis. But again, you have to be conscious of this double dip problem, right? Or is somehow having those raw p-values, can this be used to avoid that problem? I'm not sure.
Right. So that's the question that's up to debate. I think if you're me, I'm interested in finding biomarkers, and so when when I have these genes, I often just take these and run with it. People obviously want to build model and see, okay, how predictive are these genes? I think to be safe, and like I said earlier, to make reviewers happy, especially those who are in the field of machine learning. They really trying to avoid this double dipping issue, and so they would try to find an independent data set to build the model on just these selected features, and hopefully it becomes a good model.
And so with a feature selection algorithm like Relief, which is sensitive to feature interactions, on the one hand, you could expect to recover more features than from an algorithm that doesn't, because maybe some significance of some gene is masked by its interaction. You could also maybe expect the other way, like if the effect of some gene is purely, or the difference in of the expression in some gene is purely due to some other gene. So let's say you have a gene that encodes a transcription factor, and that transcription factor activates some other gene. But if it's purely the first gene, which is directly linked to the phenotype, or maybe through some other genes, but most of the genes are just co-activated by this transcription factor, then would STIR be able to actually not include those spurious genes in the result, do you think?
I think STIR would be able to detect those pure effects as you frame it. And once again, that comes back to the issue of how many nearest neighbors, I think, you want to include in your algorithm. And what we have found is that STIR with multi-serve has been working quite well in detecting both main and an interaction effect. And so if you compare STIR to, say, t-test, then I think in one of the figures in the papers, I did show that, of course, it depends on what p-values you use as a cutoff, but its precision and recall are very similar to a t-test with only main effect simulated in the data. So is that what you mean by pure effect?
No, I meant the case where the main effect is actually a consequence of the interaction effect. So a gene has no differential, well, it is differential expressed between two conditions, right? But only because of its interaction with some other gene that is sort of genuinely differentially expressed. But this gene, like it could be used as a marker.
I see.
But it's only through like a proxy of another gene.
I see. Oh, yeah. Okay. So yes, that's the point of STIR. That's what, and actually in the simulation that we conducted in the paper, it's that kind of interaction. So the simulation was done so that there's, in the case of interaction, there's no main effect. And so all of these genes are permuted in a way that the main effect is actually the same, but the effect comes from its interacting with other genes.
Yeah, I think that's still not quite what I meant. So what I would like to know is that can STIR actually detect fewer genes instead of more genes, right? Because interaction effects, they typically expand your set of interesting genes, because genes could be interesting either because they're main effects or interaction effects.
Right.
But in some cases, you would like to actually shrink your set of interesting genes, because if something is interesting only because it's a direct consequence of another gene. Yeah, I guess this is an interaction effect. It's just when you move to causality. Right, it's caused by another gene, but I suppose that's exactly what you're interested in here. So maybe I'm not explaining this well.
I'm not sure how you want to shrink this set of genes, but I mean, if you want to detect genes with just pure main effect, then I guess you can compute, which is, I think, sort of what we did with the real data simulation. But so we found about 32 genes. So STIR found 32 genes that were associated with the disease status, in this case, major depressive disorder. And but had we done t-test only, t-test found 8 genes that were associated with the outcome. And so you can look at the overlap and say, okay, well, I guess these 8 genes, the effect that STIR detected is probably from main effect. And so the remaining 24 genes, these probably contain a lot of interaction.
So one interesting word you use in the paper, and it's used in the papers you cite as well, is myopic. Could you explain what you mean by myopic in the context of feature selection?
Yes. So I didn't come up with this term, just so you know, but it has been used to describe in other papers to describe the fact that it's as the number of nearest neighbors increases, the algorithms becomes more myopic. So it's able to detect more main effect. The way that I think about it, instead of looking at all these interaction and the genes, all the genes in your data set, you're sort of narrowing down your view between just one gene and its direct connection to the outcome. And so it's becoming more myopic as the number of nearest neighbor increases.
Right. And so we discussed the possibility to apply some downstream model after STIR, but you could also just look at the p-values calculated by STIR and declare some of the genes sort of significant and differentially expressed. So that's very interesting, right? Because usually, people design this very sophisticated models based on, I don't know, negative binomial distribution or something, and some, generalized linear models. And here you can produce a p-value just by, in this almost very non-parametric way, although you still use the t-distribution. A permutation test will give you the non-parametric p-value. But still, I was curious how you can use this very, I don't know, what was the right word, very simple approach without any complicated statistics and still get reasonable p-values. What do you think about this?
I think that's exactly why I want to do something like this, in the sense that we don't have to write out each very particular interaction term, in which case it would comes back to the issue of explosion in the number of tests, but also, sorry, I just lost my train of thought. But also, do it in a particularly sufficient manner so that we have, we don't have to write out all these 2-way or 3-way interactions, but can still compute by exploiting compute the p-value by exploiting the distance matrix. Really, I think it all comes down to that. Yeah, to summarize, basically I'm just trying to rank the importance of each feature separately, almost like a univariate method, but it depends on its neighbors, on the sample's neighbors. And so it's not quite a univariate method, it's multivariate. And yeah, so we almost wanting to find a way to, because like you said, relief is a non-parametric method, to compute some sort of p-values, not retrospectively, it's easy, but to get an actual real distribution with some parametric specificities or characteristics to it, it's not quite so straightforward. But yes, with STIR, you could very efficiently compute the scores along with the p-values and take that and run.
Yeah, I think the downside of that is that it's hard to formulate exactly the null hypothesis that you're rejecting.
That's exactly right. Yeah.
You could, of course, say that my null hypothesis is that, all these like complicated mean differences adjusted and all that stuff, that they are distributed with the same mean. But that's, if a biologist looks at that, that's not very useful. It's very complicated. On the other hand, if you try to distill it to something, easily digestible, like you say, like my null hypothesis is that these genes either have like main effects or interaction effects, and it's not as obvious to draw a link between this null hypothesis and maybe your test.
Right, you're correct. I think though that you made a very good description of the algorithm earlier when you mentioned the inter differences and the inter differences, and so we could to explain it to someone who doesn't know much about machine learning, then we can describe it in the sense that the null hypothesis would be there's no, I'm going to say differences like 10 times now, but there's no difference between differences among the interclass differences and the interclass differences.
Yeah, and that would be fine, except that ignores the whole nearest neighbor thing, right? So where the interactions actually come in, because if you're just talking about interclass and interclass, then there are no interactions there. And once you get also interactions through these nearest neighbors, then it's even more complicated.
Yes, you're completely correct. Yeah, so we want to, specify that these inter and inter are coming from the neighborhood that you're defining. But yeah, it is difficult. I have to admit, it is difficult to interpret these results and try to explain it to someone. But I'm hoping that we're gonna get to that point in the future when we can, say that okay, maybe we don't know exactly where the interaction comes from, but perhaps with more experiments and analysis, then we can say, okay, we're pretty sure that these two genes interacting with each other and that affecting. Actually, there's this study that I'm trying to write up that looks at Alzheimer's diseases and actually including gene expression study, results between gene expression studies and also a GWAS study, and we're trying to do some sort of we were trying to make the connection between particular genes that people have found, but looking at it in an interaction way. But that's slightly different because we have a prior knowledge per se. We look at these three genes and some SNPs that are associated with these, and this is what we saw in the data. So that's a little bit different than what we're doing now because we don't have any prior hypotheses for STIR. But perhaps with STIR, it could produce these, dare I say, novel interacting genes that may associate with your outcome. And then you can use that, you can further confirm or verify that result in a different data set. And so I think that would prove valuable. One last thing I want to say is, once again, the STIR package is online on GitHub. And if you're interested, the paper should be out very soon, the journal published paper. And I welcome any contribution and any comments that you may have for the study.
Cool, yeah, and we'll put all the links in the show notes. Trang, that was a lot of fun. Thank you so much for being part of the podcast.
Thank you for having me. It's been a pleasure.